Hello, welcome to the Chapter 8 notes. Just a reminder, you should have your Chapter 8 reading guide out while I go through these. You can be filling that in. Chapter 8 is on metabolism, and we really focus our attention on enzymes and the different types of reactions that happen inside of cells. So, within a cell, it's basically a miniature factory. There's all kinds of reactions that are occurring, and there's conversions of energy that have to happen in many ways. Even there's organisms out there that are converting energy into light because it's called bioluminescence. But we're going to focus on metabolism. An organism's metabolism transforms matter and energy, but it is subject to the laws of thermodynamics. So I will review those in a bit here. First of all, what is metabolism? You can think of metabolism as all of the reactions that happen within an organism, and that could be within a single-celled organism or a multicellular organism. And this arises from the interactions between molecules, and your cells are very orderly, and they try to control some of this. But metabolism, again, just the totality of all those reactions. All right, so our cells have metabolic pathways, and we try to organize these. And you can see if we have a starting molecule that there could be a chain reaction here. One thing I want you to notice about this is that each one has a different enzyme. 1, 2, 3, right? And that is important because remember, enzymes are proteins, and proteins are very specific. And what makes them specific is their shape, their configuration. Their shape is everything, and their shape is specific to a specific reaction. So you can see these reactions are different, and the enzymes are different. That is very important. They are catalyzed by a specific enzyme, and enzymes are specific due to their shape. All right, a couple different types of pathways. The first is catabolic, and you might think this is a little bit funny, but I'm going to say it anyways. I don't like cats. So here's how I remember catabolic. Cats break things. That's how I remember catabolic. I don't like cats. Cats break things. So catabolic pathways break things down. That's how I remember catabolic. These processes release energy. And an example could be cellular respiration. These pathways use hydrolysis is another important point. All right, and the opposite of that would be considered anabolic. Sometimes these are called biosynthetic pathways. So if it's synthetic, we would use dehydration synthesis. We're building molecules here. These consume energy and eventually would store energy in their polymers that are being built. An example could be building a protein from amino acids. Okay. How I remember this, I'm a sports guy. Anabolic steroids, when I was growing up, was a big story. Lots of baseball players in particular were being caught using steroids. And what we call them is anabolic steroids. And anabolic steroids build muscles. So for me, being a sports guy, being familiar with anabolic steroids, it made sense that if anabolic steroids built things, anabolic pathways also built things. So that's how I remember that. I hope that helps you. All right, energy. Energy is the capacity to cause change, and it can come in lots of different forms. But what the important thing is that cells need to be able to use energy in order to do work. Cells have to be able to do things, and different parts of them have different functions, but each one has to be able to do work. So cells can transform energy from one form to another. So a couple different types of energy. You can have kinetic energy, which is associated with motion, is the energy of motion. So for instance, wind is kinetic energy. Heat is kinetic energy because it's thermal energy. Potential energy, I think of this as like the boulder on a cliff. A boulder on the middle of a football field doesn't have a whole lot of energy, but a boulder on the edge of a cliff has a ton of potential energy. So this is really stored in the location of it, could be in the structure of it, and we'll talk about an example of that with like ATP in a little bit later in this chapter. This also can include chemical energy stored in, again, molecular structure is important. So I'm going to go through a specific example of how energy is being converted from one form to another. So if we look at this picture, what we have is a boy that would climb the stairs on a diving platform. As he's climbing this, he's releasing chemical energy that was stored in his cells from the food he ate that day at lunch. The kinetic energy of his muscle movement, again, that kinetic energy being movement, so the kinetic energy of his muscle movement is converted into the potential energy as he climbs higher. More potential energy, again, because he's higher up in the air. That potential energy is in that location. As he dives eventually, the potential energy would then be converted back into kinetic energy because he's moving. Kinetic energy is transferred to the water as he enters it, and some energy would be converted to heat due to the friction of his body moving through that water. So again, just as some examples of how energy can be converted from one form to another. So that leads us to the laws that really kind of set this up. We look at the laws of thermodynamics. And when we look at this, the study of thermodynamics looks at energy transformations. So let me just explain two things. One is a closed system. A closed system would be isolated from its surroundings. This is what companies like Yeti and Hydro Flask are trying to do with their water bottles, right? They're trying to keep things cold or keep things hot. They're trying to create a closed system. It's isolated. It's trying to keep that energy inside of it. Versus an open system where energy and matter can be transferred with their surroundings. An organism like us we are open systems. We take energy in from our surroundings, we release waste. Okay, so we are open systems. So it's important to be aware of those two things, closed versus open systems. Now, the first law of thermodynamics is probably one I bet you've heard of, even if you don't know of it as the first law. It is that energy cannot be created or destroyed. It can only change forms, is usually how it's said. Or it can only be transferred or transformed. So again, energy cannot be created or destroyed. It can only be transferred or transformed. So an example of that, plants don't actually produce energy. They are transforming light energy into chemical energy. That would be an example. Another example would be the chemical energy in our food can be transferred to kinetic energy of movement of our muscles. So again, that's a energy transformation. We aren't creating the energy, we are transferring it. We're transforming it. The second law is significantly more difficult to understand. So it deals with this concept of entropy. And energy transfers and transformations make the universe more disordered due to the loss of usable energy as heat. Entropy is essentially the measure of disorder or randomness. So heat is a great example of that. Heat is extremely disordered. It's extremely random. And the more random a collection of matter, the greater the entropy. So the hotter it gets, usually the entropy goes up as well. So that leads us to the second law of thermodynamics. And the second law of thermodynamics says that spontaneous change that do not require outside energy increase entropy or disorder of the universe. So in other words, basically, for every energy transformation, every reaction, in other words, we are increasing the disorder of the universe. So every reaction is increasing the disorder of the universe. For example, the disorder is added to the cheetah's surroundings in the form of heat and in the small molecules that are byproducts of its metabolism. So everything that we do is increasing disorder. So I always like to explain this with talking about a bedroom. So how do we, as organisms avoid becoming really disorders? If everything that we do is leading to disorder, why am I so organized? I have organs, I have organelles in my cells. I am extremely organized as an organism. Well, in order to avoid disorder, which could cause death to me, what I need to do is I need to take in energy. Okay, so I need to have a constant influx of energy in order to avoid disorder to me as an organism. Okay, so I like to compare this to a bedroom. If you don't do anything to your bedroom over time, what happens? Well, it's pretty obvious what would happen to a bedroom that you don't put any energy into. It's going to become extremely disorganized, disorder, clothes everywhere, maybe. Okay, and that's when you get yelled at by your mom. Well, how do you create order again? What do you have to do? You have to clean it, right? Well, what does cleaning require? It requires you to have some influx of energy. You have to put some energy into your room. Okay, so again, in order to avoid the second law of thermodynamics as organisms, we need a constant influx of energy. So, how does that still not break the second law, you might be wondering? Well, it doesn't break it because what are we constantly giving off as organisms? We are constantly giving off heat. And that heat is still contributing to the disorder of the universe. Okay, so that's how we technically aren't breaking that second law of thermodynamics by becoming more organized, because we are constantly giving off more heat. This is a concept that I will review next week in class, so don't be incredibly intimidated by it quite yet. Again, it can be a difficult concept to master, but we will practice with it next week in class, so don't freak out too much. Living systems increase the entropy of the universe. They have to, because we can't violate that second law. And we use energy to maintain order. So I'll give you 30 seconds to read through this comic now. This is one of those comics that you can laugh at when you're educated in science. All right, moving on. Next section in chapter 8, free energy change of a reaction tells us whether this was spontaneous or not. We're not going to so much worry about spontaneity and whether something is spontaneous or not. I'm going to focus more on the energy side of this. So how can we determine which reactions occur spontaneously and which one required input? What we do is we measure Gibbs free energy. So let me grab my pen here. This is the formula for that. Actually, I think it's on the next page. Let me just see if I can get there. Sorry. Yeah, here it is. All right, so G is Gibbs free energy, the amount of free energy. Now, this free energy, what is it? It's energy that we can use, then our cells can use to do work. Okay, so there's multiple variables to this. One is enthalpy, so energy. But then we have to subtract entropy, which again is disorder, and T, which is temperature. Now, one thing that you should realize by looking at that formula is what amplifies entropy, this down here, right? What amplifies the entropy? Remember, entropy is S. So what amplifies it? Temperature. And that, let's make sense of that. Should make sense to you that there's going to be more disorder at a higher temperature. Okay, because molecules are moving around like crazy at higher temperatures. So not all energy in the system is available to do work, because some of it has to be lost due to entropy. So that disorder piece, and because otherwise it would violate that second law of thermodynamics. So the change in energy, the delta G, so that triangle we would call that delta. Delta G, what it stands for is essentially the change. So the change in Gibbs free energy during a biological process is related directly to the enthalpy change and the change in entropy. So how do we figure out how much energy is available to us? We take energy and then subtract that disorder piece, that entropy piece. Okay, and that essentially becomes what we can use. So organisms live in an expensive free energy. During a spontaneous change, free energy would decrease and the stability of the system would increase. So if we want to become more organized, is what this is saying, we have to use energy. So our energy is going to decrease, but we're going to become more organized. So delta G must be negative for that. And that'll lead me to this. All right. At maximum stability, a system is at equilibrium. However, what you need to know is as an organism, if we have a delta G, a Gibbs free energy, a free energy of 0, we have a system that can do no work. And if you as a system can do no work, you die. Okay? A system, again, that has no energy available to it can do no work and therefore cannot survive any longer. So we have to be constantly moving towards equilibrium because then we can do work. If we're at equilibrium, there's no work that can happen, and that doesn't work for a system anymore. All right. So chemical reactions being classified as either exergonic or endergonic. That's the next concept that we're going to go into. These might be terms that you talked about in chemistry, not positive on that. So here's what I wanted you to think about it before I tell you what they are. Remembering when we learned about endocytosis, which was taking things into a cell, and exocytosis, which was secreting it out of the cell, use those prefixes maybe to try to figure out what exergonic and endergonic mean. Think about that for a second. What does it mean to be exergonic as a reaction versus endergonic as a reaction? All right, so here's what the answer is. Exergonic reactions result in a net release of energy. Now, here's how I think of this. Exit. What is energy doing? It is exiting. It is being released. Okay, so if we look at this graph, it does a nice job. We have reactions, reactants, and products. So if the products have less energy, remember, energy cannot be created or destroyed. It has to go somewhere. So if the products have less energy, what happened? Well, we had to have released some energy. This is allowing that first law of thermodynamics again. Energy has to go somewhere. It has to be transferred or transformed. So if, again, our products have less than our reactants did, then energy went somewhere. In this case, it was released. On the opposite of that, an endergonic reaction would absorb energy or store. Sometimes we would store energy. Let me go back. I just want to add one thing. And a good example of an exergonic reaction would be cellular respiration. This is how we use our energy. So energy would be released from that. Endergonic, again, we have energy being stored. How do I know? Well, our reactants had less energy than our products did. So again, where did that energy come from? Well, energy had to have been inputted into it and then stored in the product. So energy was added and stored in the product in this case. Good example of this, photosynthesis. Photosynthesis stores the sun's energy in chemical energy of glucose or sugars. All right. I like these models here. This is a closed system. So remember, a closed system, nothing can go in and nothing can go out. Well, if you see here, it can do work for a little bit, but once it reaches equilibrium, there's no work that can happen here. Now, why does that not work for living organisms? Because Once you reach delta G equals 0, there's no energy anymore. The light bulb's off. You can see here there's no energy, and if you have no energy to do work, your cells die. So the cells in our body have to have a constant flow of materials. You have to have a constant flow of energy in, and then you have to have a release of energy as well. This would be like waste. This would be like the food you eat. Okay? Keeping that light on, allowing that water to spin the wheel so that we can have energy to do work in our cells. We have to have a constant influx, that's your food. You have to have a constant efflux, which is your waste. Okay, so again, this is important because delta G can't be 0. Your cells have to be able to do work. So you have to have that constant influx of energy in order for that to happen. So an analogy, this is if this was cellular respiration, which I know we haven't talked about yet in class, but if this was a cellular respiration is a multi-step process. It involves multiple steps. So each of these would be considered a step. And at each step, we can create energy. So if this was glycolysis, the Krebs cycle, and then ETC, each one of them is creating energy in the form of ATP. Now, it's also important to point out that sunlight is what provides the daily source of free energy for photosynthetic organisms and us as non-photosynthetic organisms. We depend on the transfer of free energy from those photosynthetic organisms in the form of organic molecules like glucose. In other words, it's in our food. All right, next section is all about ATP. ATP is what we would call the free energy carrier in a cell. A cell must be able to do 3 kinds of work: mechanical work, transport, or chemical. I'll go through each one of those. Example of a mechanical could be like a muscle contraction. Transport could be like active transport, moving things across the membrane from low to high. Chemical could be an endergonic reaction, like the synthesis of a polymer, for instance. So energy coupling would be a part of this. It's a feature that in the way cells manage their energy resources. So what that means is we can use exergonic processes, runs that release energy, to drive an endergonic one, which would later store it. And one of the things that we use for that would be ATP. So ATP is adenosine triphosphate. If you look at the structure of this, we actually would consider this to be a nucleotide. It's a modified nucleotide. We have phosphates, here's your sugar, and here's your nitrogenous base. In this case, it's adenine. So structure-wise, it's actually considered a nucleotide. Again, it's the cell's energy shuttle. In other words, it's the free energy carrier in a cell. This is what cells can use. It's like currency. So energy is released from ATP when that last phosphate, the third one, so 1, 2, 3, this is the terminal one, that third one, when that bond is broken there, you can see that that last one has fallen off here. That releases energy. Okay. Those phosphate bonds are really unstable, so that's what makes ATP really good at this. This bond doesn't really even want to be there, so that doesn't take a whole lot to break it, and it releases a lot of energy when it does. So what can that do? It drives an endergonic process. What does that mean? An endergonic process, remember, is one that stores energy. So ATP is going to release that energy, and that energy can then be used and stored in a different molecule. So one of the ways it does that is by phosphorylation. So that means simply transferring that phosphate. So if I go back, this phosphate that broke off is going to be transferred to a different molecule. That's what phosphorylation is. So there's 3 really types of cellular work that are powered by hydrolysis of ATP. And those are the 3 that I went through a couple slides ago. So that first one, mechanical work. This is a muscle movement. So we have a motor protein that's going to be phosphorylated by that ATP, and then it can move. So you can see this part, it moved along there. What was that? How did it do that? By being phosphorylated. ATP provided the energy to do that. Again, for this one, we have that transports. This is your cell membrane. This is active transport. So we're moving low to high against the concentration gradient. ATP is going to phosphorylate this protein, which changes the shape. And then this last one, what it's doing is a more chemical approach. And in this case, it's building, and this is an amino acid that got built by phosphorylation. So each one of those ATP supplied the energy that allowed that to happen. Whether it was moving a muscle, moving a protein on the membrane, or making an amino acid, it was ATP that supplied the energy to do that. Now, what does ATP become when it breaks off that last phosphate? It becomes ADP. So adenosine triphosphate, tri meaning 3, adenosine diphosphate, di meaning 2. So it only has the 2 there. All right. Catabolic pathways drive the regulation, or the regeneration of ATP back from ADP. So again, here's that ADP, adenosine diphosphate. So we would add that through a catabolic process. All right, one thing I just want to share with you is a crazy statistic, in my opinion. ATP, first of all, is considered a renewable resource in your cells, and that's just because it can be recharged by being rephosphorylated. This happens in a working muscle. So if, say, you're working out, maybe you're running a race, a working muscle cell recycles its entire pool of ATP once a minute. So more than 100 million ATP are consumed per second. Now, 100 million ATP are used in a working muscle cell per second. Now, 10 million might not sound like that much. One of the things about our world now, those numbers, million, billion, they get thrown around like they're nothing. And a lot of times what I believe is that we don't understand how big these numbers are. Let me just explain to you one thing. 1 million seconds is 11 and a half days. 1 million seconds is 11 and a half days. Now let's put this in perspective of how much bigger a billion is than a million. So 1 million seconds is 11.5 days. 1 billion seconds is 31 and 3/4 of a year. So almost 32 years. 1 billion seconds. 1 trillion seconds is 31,710 years. So just to put in perspective how big those numbers are, I think is important in life in general, to just understand how big that number is. All right, now I'm going to get into enzymes. Enzymes are catalysts. They are biological molecules. They are proteins that act as catalysts. What that means is they speed up chemical reactions. And what we're going to look at is how that happens. So let me just give you an example of that. The hydrolysis of table sugar, which is sucrose, which is a disaccharide, to glucose and fructose, so we basically just are breaking that disaccharide into those monosaccharides, is considered exergonic. Despite this, in other words, it can happen spontaneously. Besides this, if you didn't know, sucrose is table sugar, by the way. Despite this, your sugar sits in its bowl with no observable hydrolysis. You don't notice the breakdown of that sugar. However, if we added a small amount of an enzyme called sucrase to that solution, the entire thing could be hydrolyzed within seconds. So what that is showing, what I'm trying to show you there, is how important these enzymes are, these catalysts are, to speed things up in our reaction, in our bodies, speed up our metabolisms. So these enzymes are catalytic proteins. They regulate metabolic pathways. And every chemical reaction between molecules involves both bond breaking and bond forming. And enzymes are going to do both. They're going to both break bonds at some times and they're going to form bonds at other times. So the hydrolysis, and again, this is the example I just went through, sucrose to glucose and fructose. Sucrase is our enzyme. What did it do in this case? It broke this bond, the glycosidic linkage, right? But it added these bonds, right? It added the water, right? The OH group here and the hydrogen here. So it added those bonds. So it broke one bond, added other bonds. That's what sucrase did. Now, one of the things that we have to understand is activation energy, because this is basically Catalyst 101. How does it speed up a reaction? Well, what it does is it lowers activation energy. So what is activation energy? It's the initial amount of energy needed to start a chemical reaction. So if you think about what I just said, a catalyst lowers activation energy. Well, what did it do? It just made it easier to start the chemical reaction. So activation energy is often supplied in a normal circumstance when there's no enzyme in the form of heat. Okay. Now, let's think about this a second. Why can't we always just use heat then? Why can't we just use heat as our activation energy? Well, if you remember what we learned about proteins, they're very sensitive to temperature change. So we can't just rely on temperature always, because if we got too hot, the enzymes would break down, or all your proteins would break down. They wouldn't work. Okay. So this is a really important graph. This shows here's our reactants, here's our activation energy. Let me grab my pen here. So from here to here is our activation energy. This is what energy we need in order to get this reaction started. And once the reaction has started, here would be our products. And from here to here would be the energy that in this case was released. How do I know what was released? Well, our products have less energy than our reactants. So if that's the case, the energy had to go somewhere. In this case, it would have been released. So this we would call an exergonic reaction. All right. So an enzyme catalyzes reactions by lowering the activation energy. So now let's look at the same graph with an enzyme. So the black line was the original one. This is without an enzyme. And you can see, well, what was the difference here? If I grab my pen again, this was our original one. And grab a different color here. And this is with an enzyme catalyst. You can see activation energy was lowered. But what do we get out of it? It's the same amount of energy was still released. So the enzyme didn't lower the amount of energy that got released, and vice versa. If it would have been an endergonic reaction, it doesn't reduce the amount of energy that got stored. All it did was lower the amount of activation energy that was needed in order to get it started. So you can see why, probably, these are such important molecules in our bodies. Okay? They basically make us more efficient. All right, a couple terms that you should know. One is a substrate. So a substrate is it's basically the thing that the enzyme acts on. It's the reactant that the enzyme acts on. So, and basically what it's working on, I think of it as. The enzyme is going to bind to its substrate and form a complex. It's going to convert the substrate to its product. Again, important to re-point out here, the enzyme is specific to its substrate. Why? Because the enzyme is a protein and it has a specific shape that will fit only a certain substrate. change the substrate, it won't fit in that enzyme anymore. So here's an example. Here's our enzyme. You can see it has a specific shape where that only this substrate will fit into there. Okay? And where it actually comes in contact with the enzyme is called the active site. So where the substrate bonds to its enzyme is called its active site. And that active site has to be the perfect shape to fit that substrate. That's why, again, you change the shape via pH, via temperature. You're changing the active site. It can no longer bond to its substrate anymore. Sometimes we have what we call an induced fit, and that just means that when the substrate comes in contact with the enzyme, that enzyme slightly changes its shape. And what that would do is it just allows it to come in better contact. You can see, though, the difference between this and this. The enzyme has just slightly changed its shape. So an enzymatic reaction, the substrate will bond to the active site. It actually does do bonding. It could be like through a hydrogen or ionic bond, depending on the enzyme and the substrate. Okay, a couple things I want to point out with this diagram. One of them is that an enzyme is reusable. So once it changes its substrate into the products that are released in this case, that enzyme can go and do it again. Okay. Another one with that depending on the enzyme, it could go this way. In other words, maybe we're going to build bonds, maybe we're going to break bonds. Depending on what the body needs, different enzymes would do different things. Another thing to know about an enzyme is it does depend on the concentration of the substrate. In other words, if there's no substrate to act on, the enzyme can't do anything, right? So concentration of the substrate does matter. It also matters the concentration of the enzyme. All right. So the active site can lower activation energy. How? So how do catalysts actually lower activation energy? Maybe it's orienting the substrate the perfect way. Maybe it's straining the substrate's bonds. Maybe it's just providing a favorable environment. It could be covalently bonding. So again, that's just ways. You don't have to know these for your test or anything, but this is how it actually works to act as at lowering activation, acting as a catalyst. The activity of an enzyme is affected by general environmental factors, and this is the same factors that affect a protein in general. And the reason why is because it's all about shape. So if we looked here, we have two different enzymes. We have a typical human enzyme, and then we have a heat-tolerant bacterial enzyme, and it is adapted at its optimal temperature is almost 80 degrees Celsius, where a human enzyme is optimally at about 37 degrees Celsius, which is about 98.6 degrees Fahrenheit. So again, just showing you that different enzymes will have different optimal temperatures. And the same thing is true with pH. So here we have a stomach enzyme, which would make sense if you have to be able to work in the environment of the stomach, that it would have a low pH. And for this one, this is an intestinal enzyme, which has a much higher optimal pH. So just showing you that pH and temperature are very important. Why? Because it affects the shape of those enzymes. There's other things that are impacted, such as non-protein enzyme helpers, such as zinc, copper, iron, other coenzymes like vitamins. Okay, so some of you maybe take vitamins at home that actually can help your enzymes out. All right, this is really important, so please clue in. Almost done, but there's this last piece is pretty important. I have competitive and non-competitive inhibitors, and then I'm going to give you some examples. All right, so competitive inhibitors, what are you doing if you're competing? You're trying to get the same thing. So what in this case, they're the substrate and the inhibitor. Now, what does it mean to inhibit? I should probably include that. Inhibit means to stop. If you ever see inhibit in AP biology, think stopping it from happening. Okay, so competitive inhibitors, they're competing for the active site and they're stopping the substrate from getting in there. Okay, so they're competing with the substrate and they're stopping this process from happening. That's a competitive inhibitor. It's competing for the active site to stop it, stop that substrate. A non-competitive inhibitor, it's not competing for the active site. It's going to bond somewhere else, which I'm going to talk about more in a second here. But it's still doing what? It's still inhibiting. It's still stopping that process. So non-competitive, because it's not competing for the active site. However, it is still stopping it. So it's still inhibiting the process. All right. There's a couple terms that are going to be related to that. And these metabolic pathways are highly regulated. So one of the ways that we regulate them is through allosteric means. Allosteric is an absolutely essential term. You have to understand what allosteric means. All it means is it's bonding somewhere other than the active site. So this is non-competitive. Okay, it doesn't necessarily have to be inhibition. Allosteric just means something is bonding and regulating the enzyme away from the active site. Okay, and lots of them are regulated in this way. So let me just show you this. This would be one that actually helps an enzyme. So this is what we call an activator. It's still bonding allosterically away from the active site. Here's your active sites. It's bonding away and it's helping us stabilizing it. Versus an inhibitor, it would bind again away from the active site. However, it's going to stop the process. So if we have an activator, it's going to allow it to happen and, in fact, encourage it to happen. An inhibitor is going to stop. But both of these are allosteric because they're bonding away from the active site. Sometimes we can have what's called cooperativity. This is a form of allosteric regulation that can amplify. Again, it's going to encourage this to happen. We can have what's called feedback inhibition, which is when the end product turns it off. So once we have it, it's basically what I think of with this is it prevents the cell from wasting resources. Once we have enough, it stops the process. So the end product, in this case isoleucine, which is an amino acid, it's going to turn off the process. Once you have enough isoleucine, we don't need more. We don't want to waste resources. It's going to turn off the process. Within a cell, enzymes may be grouped into complexes by steps. For instance, these 5 enzymes might be grouped into a complex to make it more efficient. They might have a fixed locations, maybe like inside of an organelle. For instance, a mitochondria is going to have specific enzymes that are going to help to break down glucose into ATP. Metabolism, again, the set of pathways. It's all choreographed into play of thousands of reactions that happen within a molecule or within a cell. It's all choreographed by the different things in the cell, though. Very important, and we will review this next week. I know that was a lot. I would encourage you to watch this video because I know this can be a kind of an abstract lesson. So watch this. It'll be a good review. And then again, we'll review again next week. So have a good one.